Hello, class. Welcome to week 5 of Crop Physiology Lab. This week, we are starting the Image J activity. Now, we will do this Image J activity and we'll learn this new software, Image J, offered through the National Institute of Health. But the work that we'll do in this activity sheet really requires you to have mastery of the material presented in the experimental design and analysis form from week 2. So make sure that you have those Excel analyses done. Make sure you can make a bar graph, do a scatter plot, and do those calculations, calculate some means and differences. So this week, we're going to work together in Image J. We are going to have an activity sheet that's due. I will have, of course, by this point, had your journal article summary written and provided, but understand that next week, you've got some materials that you'll have to track down. You'll need baking soda, soap, syringe, leaves, a hole puncher, some clear plastic cups, and a nice little table lamp. This is for the photosynthesis and respiration lab next week in week 6. It's the first week of March. So for now, let's hop right into the Image J portion of class. So for that, I'm going to start by pulling up the activity sheet. You'll see on this activity sheet that you need a sheet of white paper with a known length and width. I'm just using a plain blank 8.5 by 11 sheet in my case. You'll also need to track down a medium or large sized leaf with partial necrosis. This will be necessary for today's activity. I found an avocado leaf that I will show you later. And this avocado leaf has a good deal of necrosis, right? It's got a good deal of dying or decayed leaf material, but also has a little bit of green. Now, this could be death through biotic or an abiotic stress. It could be, say, a pathogen, like we see in part 2 of this assignment. I think mine's mostly water stress. And then you'll need to download the wheat leaf rust picture from Blackboard. It's the same picture here. You can download it from the activity sheet, or you can download it directly from Blackboard. Okay. So this first part will require you to get that leaf material and put it on top of the sheet with known dimensions, and your leaf must fit on top of that sheet of paper. It can't be off of it. So photograph that, email that photograph to yourself, and copy and paste that raw picture here for part 1, and then the cropped picture. So what I ended up doing was taking this picture of my avocado leaf and sent this to Microsoft Paint. Just about every computer has some sort of generic, easy to use image processing system available. Microsoft Paint, I use the select tool, and I use this select tool to crop it. And it doesn't have to be a perfect crop, right? If we don't have these dimensions quite right, you'll see that we'll get a lot of overlap, in this case of the table for me, right? If I scroll down, uh-oh, that's no good, right? We don't want that. So what I'm going to recommend you do is be a little bit, dig into this paper a little bit. At the end of the day, we're really just hoping for estimates, not exact values. So start inside just a little bit. If you can see where those dashed lines are going, this is the place that we're cropping the picture to. And then scroll down and make sure that you don't have any of that backdrop. In my case, my backdrop is the table. So I don't want to have any brown wood in this picture. Just want that nice 8.5 by 11 sheet of paper. We're going to pretend like the section that I've cropped is indeed the whole 8.5 by 11 sheet of paper. Now, if we're doing this for real, we'd use fancy scanners that'll take care of this for us. But we're doing this with our cell phones, right? We're doing applied crop physiology. We were doing this all in real time. So you should see that this blue and white dashed line doesn't have any of the table in it. It's got just about all of that 8.5 by 11 sheet of paper. And inside this 8.5 by 11 sheet of paper, we've got the avocado leaf with a little bit of necrosis. Hopefully your picture looks somewhat similar. Doesn't have to look identical. Just make sure you have some brown section on here and some green section on your leaf. Okay, I'm going to copy this. You can either right click and do copy, or you can do Control-C. I'm going to go up to this file and click new. And this new one, I'm going to paste the picture that I just copied, or you can do Control-V, and you'll see that our avocado leaf on this white sheet of paper is now present. So you have cropped your picture, right? So paste that uncropped and that cropped picture in part 1. Next, you'll have to go to image J. At this point, you've had about 3-4 weeks to download image J. There's not really an excuse that you can offer me. So please have image J downloaded. And in image J, you should pull up just what you see here, this little file box that says image J after you download it. It's a pretty boring display. I don't know what to tell you. You can't make it much bigger, but what you can do is you can load that file in here. So I'm going to click File, Open, and I'm going to move this here. I'm going to pull that processed avocado. So I saved that picture in Paint. I saved it as avocado processed, as opposed to the picture with the wood table in the background. This is just the 8.5 by 11 sheet of paper with an avocado leaf in it. All right, so here's what we're looking at so far. We've got image J up. We've got our processed avocado leaf picture in here. Your leaf won't be an avocado leaf. Yours is just what you pull from outside. If you have an indoor plant with a little bit of necrosis, that's just as good. And you'll see that this leaf isn't perfectly flat. That's okay. At the end of the day, we're just doing estimations at this point, right? We're just really learning how to use image J as the software. From here, take a screenshot of this and paste it in part 2. So I can see that you've got image J in the screen, you've got your picture in the screen, and you're doing your analysis. At this point, we're going to actually do the analysis, and here's where this teaching part comes in. The first thing that we're going to do is calculate leaf area. Now, how do you think we can do that? Well, we know that this whole picture is based on an 8.5 by 11 piece of paper, right? This whole image that we're looking at measures 8.5 inches in width and 11 inches in height, right? So I'm going to write that down. Image equals 8.5 inches by 11 inches. In fact, I'll even write it on the Word document. Image starting equals 8.5 inches wide by 11 inches long, however you want to label it. So what is that in square inches? I'll just pull out the calculator. 8.5 times 11. This total image area is 93.5 square inches. All right, good stuff. So let's pull this image J material back up. We know that we're working in this 93.5 square inch image. Okay, some people will use a ruler. Let's not deal with that. We're just going to hop straight into using what we know. So I'm going to bullet that actually. Let's put a little bullet point there. Now, the next step is to figure out what percentage of this paper is a leaf, right? Including that necrotic region. So how can we do that? What we're going to do after we've uploaded our image is click Image. Then we're going to click Adjust. Then we're going to go to Color Threshold. And you'll see that this new box has popped up. Let me adjust this so you can see it well enough. Okay. On here, you'll notice that we have 3 threshold color options. We've got hue, we've got saturation, we have brightness, and we see on these graphs that there are these peaks of color. Okay. For our purposes, we're just going to keep these values set as they are, default, red, and HSB. So what are these? Hue, saturation, and brightness, these are aspects of color in the RGB, the red, blue, or red, green, blue scheme. These terms are really often used in reference to the color of each pixel in a cathode ray tube display, right? Think about like an old school TV, right? All these possible colors can be specified according to their hue, their saturation, and their brightness or brilliance. Kind of like this RG and B component is what we're really looking at. Now, most sources of visible light contain energy over bands of wavelengths, right? Hue is the wavelength within this visible light spectrum, which the energy output from a source is greatest. And here we see that it's shown as these peaks or these curves, right? Imagine these peaks as a curve to represent what we see in this picture. Now, saturation is an expression of the relative bandwidth of the visible output from a light source, right? We see a lot on this end of the spectrum for saturation for our particular example. It's represented by the steepness of a slope of curves. And we can see that this is broken down from a low to a high saturation. And of course, brightness is the one that we're all probably most familiar with. It's the relative expression of the intensity of energy output of a visible light source. So that's all well and good, but how do we apply that to this particular setting? In this case, we can use hue, saturation, and brightness to select certain parts of this piece of paper, right? If we take everything off the hue spectrum, we're just looking at the initial image, right? We're looking at the avocado leaf on an 8.5 by 11 sheet of white paper. But what happens if we start adjusting our hue, right? We see that we cover more of this sheet of paper in red, and this is important. We're going to use the red color to select certain parts of the paper. We're going to start by selecting all of it. We adjust the hue. See what happens if we adjust saturation. We have that same effect. We can adjust brightness. And by taking this brightness through this whole band, we're making sure that we have everything covered, right? From 0 to 255, 0 to 255, and 0 to 255 for hue, saturation, and brightness. Okay. Now what do? Now we need to go ahead and process. And we're going to set this as a binary, kind of a yes, no, or in this case, a black and white sort of system. I'm going to click Make Binary. So 100% of this image is now set in black because we have that whole spectrum of hue, saturation, and brightness selected. Go ahead and click Analyze, then click Measure. You should see this box pop up with results. We're given an area. Our area, I'm going to write this down. Oops, let's see, best way to do this. Our area is 1195200. I'm going to call this total area. Okay. So now we're asked to calculate the leaf area, right? So we have the whole area of our avocado leaf on the 8.5 by 11. Let's close this. Don't save. Close this color threshold. Close the image that we were working with and reopen it. File, open, processed avocado leaf. We have this box that popped up again. That's a good sign. So now let's go back to that step. Image, adjust, color threshold, and click that. And now we need to adjust our hue, saturation, and brightness so that we're only looking at the leaf. Well, how do we do that? Let's kind of click around a little bit here, see what we get. If we do something like that, maybe we see that if we do this, this is tricky, isn't it? How do we get this to work? So I recommend spending a little bit of time on this. So it's not necessarily as easy as you might hope. Let's see. Oh, okay. So what are we doing with this clicking around? We're selecting different parts based on this range of hue to start with. Then we can make these adjustments on the basis of saturation. Well, no, that's not the right way. What if we try this route? Aha. So if we decrease the range in the positive direction, the higher direction of saturation, we're seeing that we're only selecting that leaf material. That material within this section of the hue range, 0 to 82. You can probably even adjust that down. Well, it's good enough. We want to make sure that we keep that green area. We see that this line's set for the green part of the spectrum. And I think that's good enough on hue. Got 0 to 101. Saturation, oh, let's see. We want to get this as close as possible. It doesn't have to be perfect, but we want to get a good chunk of that leaf selected. I'm going to do, notice if we do this, we don't have too much effect going on. 82 to 255. And then brightness is probably actually pretty darn good. We might not have to do too much with that, but if we're lucky, we'll get a little bit off that section at the top here, which I know we have to chop off. Well, I think that's good enough for my purposes. I'm going to close that out. And I'm going to click Process and go through that same set of steps again. Process, binary, make binary. So now we're looking at it in black and white. So instead of having the whole sheet selected in black, now we're looking at just the leaf in black. We've got a paintbrush tool, which we're going to use as an eraser. I'm just going to kind of chop a little bit of that off, see if we can make this estimation a little bit more accurate. But you got to be careful with this, otherwise I'm going to end up doing more damage than need be. I think that's pretty good for our purposes. Now what do? Now we're analyzing, we're getting that measurement. So we're going to click Analyze, Measure. Notice this box pops up again. Aha, instead of saying 1195200, we've got a new area. This is our leaf area. What did that say? Our leaf area, it says, is 123,809. Okay. 123,809. And it doesn't really matter too much what these units are. These are just pixels at the end of the day. We're using these values to calculate percentages. And let's use this now, right? At this point, we know that our total area is 1195200, and our total leaf area is 123,809. So what would we do? We just divide total leaf area, leaf area by total area. So this equals 123 809. I'm just gonna, yeah, I'll put that in. Divided by, whoops, divided by 1,195,200. 1,195,200, which equals 10.4%, more or less. Okay, so how do we use this 10.4% to calculate leaf area in units of square inches or square centimeters? Well, we have a pretty good starting point here. We know that our 8.5 by 11 is about 93.5 inches squared. So all we have to do is leaf area equals what? 93.5 times 0.104, right? And that's telling us that our leaf area in units of square inches is 9.72, well, let's do 9.7 inches squared. It doesn't have to be perfect. These are just estimations. Okay, that's good. Now, what is this value in centimeters? Well, actually, let's just put this on the same line. 1 inch equals what? 1 inch times 1 inch. 1 inch is 2.54 centimeters. So 2.54 times 2.54 is 6.4516. Okay. So we can just multiply that through. 1 second. So we do 9.7 times 6.4516, and that's 62.6 centimeters squared. Okay, so we've now calculated the leaf area. I guess I'll put these images in here as well. Picture from this device. We'll decrease the size of this to make my life easier. Insert pictures from this device. Whoops, two of the same. And I'll let you put your own picture for two, of course. I don't need to upload that, but that's just a screenshot for section 2. Okay, so we did parts, or the answers to 1, 2, and 3 with my example, not what you're going to calculate, but with my practice round for the avocado leaf. Now we need to calculate the percentage of necrotic leaf material and report it in units of square centimeters and square inches. So let's pull image J back up. We know that we need to close all of this out. Don't save the measurements. Don't save the picture. We're going to do File, Open. Again, we're going to pull up avocado processed, right? That picture that we processed and cropped so that we don't have any of that outside material. And this is, we're making the assumption on an area the size of 11 inches long and 8.5 inches wide. Okay, so you know the drill. Now we're going to click Image, Adjust, Color Threshold. And we're trying to, we have already leaf area. Now we want to calculate the leaf area that's green, right? Or we can calculate the leaf area that's brown, but the leaf area that's green is going to be easier for us because we want to figure out the necrotic region, right? You can take whatever approach to this that you want, but that's how I'm going to do it. I'm going to calculate that green section. So let's do, how do we do this? Tricky, tricky, isn't it? Oh, we have a start. Okay. So we adjusted the brightness to start this time. Now, why would we do that? That's because most of this paper is white, right? So by setting up the brightness accordingly, we can focus on the leaf. Saturation, not helping us too much here. Oh, now we just played with the hue a little bit more. And I think we're looking at a pretty good estimation of that green area on the leaf. So you see the green area is now turned red. And from here, we can set this as a binary, that black and white image. So we started by adjusting the hue to avoid a lot of that necrotic region. We adjusted the brightness to avoid the paper and the saturation, right? So let's go with the next steps. We're going to go process, binary, make binary. That looks like a pretty good leaf to me. I'm happy with that. I click analyze, measure. And we see this box that pops up again now. This is 24228. Okay, so I'm going to bring my leaf area over. We'll just do a copy and paste here. We know that our total leaf area is 9.7 square inches, or 62.6 centimeters squared. We know that our pixel area is 124,000. Total area doesn't matter too much at this point. So we saw that our green area of the leaf, this section here, leaf green area as opposed to leaf brown area, equals 24,228. All right, so what do we need to do at this point? We can calculate a percentage of green and brown area. And how would we do that? That'd be 24,228 divided by 123,000. So that's about what, 20-ish%? Let's see what my calculator says. 24,228 divided by 123,809. That's 19.6%. Okay, let's calculate the percentage brown area. Well, that's just total area of the leaf is 100% minus 19.6% equals what? 80.4%. Okay, so we know our percent of green area and our percent of brown area. We're going to calculate the percentage of necrotic leaf material. That's the brown area. So necrotic area is 80.4% of the leaf. So now if we're doing 80.4%, and we have our leaf area already, we can say which makes, which makes up 9.7 times 0.804, 7.8 inches squared, or what? We can do that same calculation for centimeters, right? 62.6, that's the value that we already calculated for leaf area, times 0.804, which is 50.3 centimeters squared. Now, do these values make sense? I think so. 7.8, it sounds like it could be about 80% of a total leaf area of 9.7 square inches. Same with centimeters squared, right? So you'll need to do the same activity, but with your own leaf picture, right? So go out, find a picture of a leaf, or find a leaf with some good necrosis on it. Could be small circles scattered across the leaf, whatever works best for you. And then upload it on your computer. Go ahead and crop it, upload it to image J, paste a screenshot of that. That's just using the screenshot button on your computer. Then calculate the leaf area and calculate the necrotic area. You'll see that there's a part 2 to this. The use of part 2 is more of an applied use, right? It gives a quick background. Wheat breeders need to develop varieties with good disease resistance. Makes sense, right? One of the major diseases of wheat is leaf rust, and breeders routinely screen for resistance to this pathogen. Sometimes you'll see it in the field if you want adult plant resistance, or in a greenhouse if you want single gene resistance. But you can look at the leaf and say, well, this has more disease than that, or we can use image J to calculate the percent of the leaf covered with leaf rust. And we can do this 1 day after infection, 4 days, 6 days, and 7 days after infection. Now, I want you, as the wheat breeding assistant, to use image J to calculate the percentage of leaf covered in leaf rust at each of the 4 days after infection for the variety Deccan and Retro. So, right, we have 2 varieties and 4 days after infection. You'll need to do the calculations for each of these. What percentage of this leaf is covered in leaf rust? Say Retro 2 days after infection, 4 days after infection, 6 and 7. Do the same for the other variety, right? So you should have a bar graph that has a total of 8 bars on it. And you're only presenting means. You're not going to present standard deviation because we don't have replications. So there should be a bar graph for each variety that shows percentage of leaf covered in leaf rust at each of the 4 days. If you're very clever, you'll figure out how to do this sort of work without breaking it into 8 total images. But the easiest and most straightforward way from what I showed you is to break this into 8 images. Save, for instance, this leaf as Retro 7 days after infection, Retro 6 days after infection, and so forth, and calculate the percent of leaf covered in leaf rust. I don't need this to be in square inches or square centimeters, only present it as a percentage because we don't know the size, the initial size of this image. All right, with that, I look forward to seeing your results. Let's pull up the schedule. Reminder, get your materials for next week. We'll have an activity sheet for next week, but please complete this activity sheet by the end of the week. And I will talk to you all next week. Thank you so much.